Welcome everyone. Today we will be discussing the different types of chemical bonds that link monomers in a polymer and their relation to secondary structures. Let's get started. Covalent bonds between monomers in a polymer are particularly strong, allowing them to be connected in a repeating pattern to form lengthy chains. The magnitude and power of these bonds vary depending on the monomer type. Even the same kind of monomer can be linked by different types of covalent bonds, thus creating numerous kinds of polymers. A peptide bond is the type of bond that holds the monomers together in a polypeptide or protein. This bond is formed when the carboxyl, COOH, group of one amino acid combines with the amino, NH2, group of the next amino acid, releasing a water molecule which is known as dehydration. The peptide bond is essential for the protein's structure stability and the biological process it is associated with. Glycosidic bond is a crucial structure in biochemistry, formed when two monosaccharides join together, usually through the removal of a water molecule. This bond is also vital in creating polysaccharides, a chain of connected monosaccharides. The process of developing this bond is known as the dehydration process. This bond is responsible for the complexity and diversity of living organisms and is integral for biochemistry. Biomolecules are the building blocks of all living organisms, and nucleotides are a particularly important type. An ester bond, formed between the phosphate and hydroxyl group of sugars, is a key characteristic of nucleotide structure and its significance cannot be overstated. This bond is integral for the transmission of information within living organisms and is found in all life forms, thus making it a basic part of life. Biomolecules are essential components of all living organisms. Proteins form the framework of cells and nucleic acids are responsible for genetic inheritance. A prime example of how biomolecules are used is through the phosphodiester bond. This bond is created by the phosphate moiety connecting the 3' carbon of one sugar of one nucleotide to the 5' carbon of the sugar of the subsequent nucleotide, and held together by ester bonds on either side of the phosphate. This structure is needed for the proper functioning of nucleic acids within living cells. The Watson-Crick model is a well-known example of a secondary structure exhibited by DNA, suggesting that DNA forms a double helix structure, the most commonly known shape of nucleic acids. However, nucleic acids can also take many other secondary formations which vary in complexity. Biomolecules are essential to life, forming the basis of its structure. Polynucleotides, two strands running in opposite directions and connected by sugars and phosphates, form the backbone. Nitrogen bases are then projected onto the backbone, facing inwards. The combination of strands and bases creates the foundation needed for life to exist. Biomolecules are truly amazing and can be a fascinating subject to explore. Two strands of nucleotides, with adenine linking to thymine and guanine linking to cytosine, are held together by 2 and 3 hydrogen bonds respectively. This pattern of base pairing is essential for life and exploring its fundamentals can be a very exciting and rewarding experience. Biomolecules are a complex compound of organic molecules which are essential for life. They appear like a helical staircase, where each step of ascent is formed by a pair of organic bases. One full turn of the helical strand consists of 10 steps or 10 base pairs. This helix of organic molecules rotates 36 degrees at each step of ascent, and this structure is seen universally in biology, being vital for life. bDNA is the most common and widely studied form of DNA in biological systems, characterized by a pitch of 34 angstroms per base pair and a rise of 3.4 angstroms per base pair. This specific arrangement is essential for the highly accurate replication of genetic information. DNA and RNA are distinct molecules, each with its own set of components that make them unique. DNA contains adenine, thymine, guanine and cytosine, whereas RNA has adenine, uracil, guanine and cytosine. Additionally, RNA has fewer oxygen atoms than DNA. Both biomolecules are essential building blocks of life. Biomolecules are composed of smaller molecules, known as monosaccharides, and are held together by different types of chemical bonds. Peptide bonds are formed between two amino acids, hydrogen bonds are formed between two molecules that have partial charges, glycosidic bonds are formed between two monosaccharides, and covalent bonds are formed through the sharing of electrons between two atoms. Glycosidic bond is a covalent bond between two carbohydrate molecules, where one molecule provides an oxygen atom and the other provides a hydrogen atom. It is vital for production of many biological molecules, which include DNA and proteins. The formation of glycosidic bond occurs through a process called glycolysis. This process involves the interaction between oxidation and dehydration, where water is removed from either one or both of the molecules. Then oxygen and hydrogen atoms combine to form the glycosidic bond. Additionally, it can also be formed through esterification or hydration. Two molecules combining and exchanging positive and negative charges form an ester bond. In this slide, four molecules, a phosphate, a nitrogen base, a phosphate group and a hydroxyl group, together form an ester bond. The combination of the phosphate and sugar creates an ester bond, as does the combination of the nitrogen base and sugar, and the phosphate and phosphate group. Additionally, the hydroxyl group and the ribose together form an ester bond. Biomolecules like proteins, carbohydrates, and lipids are the fundamental building blocks of life. In this presentation, we take a closer look at our body's components in their dynamic state and learn about metabolism. Metabolism is the process of chemical reactions that convert the energy from the food we eat into molecules and energy that our bodies can use. Knowing how this works is essential for good health. Biomolecules are crucial for the existence of all living organisms, ranging from bacteria to plants and animals. Quantified as moles per cell or moles per liter, these molecules are indispensable in enabling cells to perform their necessary metabolic processes. Without their presence, life as we know it would not be possible. Biomolecules are fundamental to life, and a thorough recognition of their significance is a necessary step in discovering the mysteries of biology. One of the incredible traits of biomolecules is their constant breakdown and formation. Metabolism is the process through which these molecules are regularly broken down and afterwards regenerated in order to form new molecules, making up a perpetual movement. This indispensable process is indispensable for all living organisms. A deep appreciation of the turnover of biomolecules is indispensable in continuing our knowledge of the living world. Biomolecules are integral to several metabolic processes, one of which is decarboxylation. This process entails the removal of carbon dioxide from a molecule, often an amino acid, and its transformation into an amine. This process plays a major role in the metabolism of a healthy body. Biomolecules play a major role in the environment. An illustration of hydrolysis can be seen on this slide. Hydrolysis is a chemical reaction that uses water to break the molecular bond. Knowing how this process works can help improve our knowledge of the environment and the connections between different elements. Compounds containing an amine group can form biomolecules such as proteins and nucleic acids. Deamination is the process of removing this group from the molecule, as illustrated in the picture. Metabolites, essential compounds for life, are transformed into each other in a metabolic pathway. These pathways can be compared to the automobile traffic of a city, having junctions and intersections that transport metabolites at a certain speed and in a certain direction. Metabolic pathways are essential for the effective flow of metabolites, which are key to life. Biomolecules are the essential components of life, and metabolism is a system of reactions that plays a critical role in preserving the body's functioning state. Metabolite flow is the continuous shift of bodily parts which is integral to our well-being. An even, unified metabolic pathway is necessary for our physiological activities to flow without impediment. Generally, metabolic reactions are not singular, but rather intertwined with other reactions as part of this intricate system. Biomolecules play a key role in metabolic conversions in living systems. They increase the speed of reactions, thus happening much faster than they would in the absence of them. Carbon dioxide, which is a physical process, is made possible thanks to catalyzed reactions in organisms. These metabolic conversions would not be feasible if biomolecules were not present. Biomolecules are essential components for the construction of living organisms, primarily composed of a variety of elements. Manipulating these elements can generate energy and carry out essential functions in the organism. This slide features 4 distinct ways of eliminating CO2 from molecules: dehydration, hydrolysis, decarboxylation, and esterification. Each of these methods has its own pros and cons, so it is essential to have an understanding of the respective biomolecule to determine which one is appropriate for a specific application. Biomolecules are fundamental for life and enable multiple crucial activities. Deamination is a process that has an effect on biomolecules, involving the removal or addition of an amino group, a water molecule, or both. This transformation can affect the characteristics and purpose of the molecule. Biomolecules like proteins, DNA, and lipids play a huge role in our biological structures. To precisely measure and regulate their concentrations, various units are used, including mg, cells, GMS, cells, micrograms, cells, moles, cell, and moles, liter. Gaining knowledge on these units is vital to accurately measure and manipulate biomolecule concentrations. Biomolecules are at the heart of life, playing an essential role in sustaining living organisms. The 4 major classes of biomolecules are carbohydrates, lipids, proteins, and nucleic acids, which interact with each other to drive the biological processes that characterize cells. Metabolism is the set of chemical reactions responsible for the production and utilization of energy in the living state. This presentation will explore the intricate metabolic pathways in living organisms. Biomolecules are fundamental for life to exist. Metabolic pathways allow complex structures to be formed from simpler ones. For instance, glucose is produced from water and carbon dioxide in mesophyll. Additionally, lactic acid is generated from glucose in muscle cells, and cholesterol is produced from precursors like acetyl-CoA. These metabolic reactions are fundamental for living organisms to develop, renew, and maintain their bodies. Discussing the breakdown and assembly of biomolecules, and the energy implications of these processes, we can observe how degradative pathways lead to the release of energy, while biosynthetic pathways consume energy. Gaining insight into these processes can help us use them to our practical advantage. Glycolysis is a metabolic pathway that plays a key role in all living organisms. This pathway is utilized to break down glucose molecules into 2-pyruvate molecules, releasing energy in the form of adenosine triphosphate, ATP. This energy is then used to support a variety of biochemical and physiological processes, such as protein synthesis and the production of essential metabolites like lipids and fatty acids. Glycolysis is critical for energy production and is essential for a range of cellular activities, making it an important factor in maintaining homeostasis in living systems. Biomolecules play a crucial role in the functioning of all living organisms. An intricate system of these chemicals works together to keep the body alive and functioning. Glucose, for instance, is present in a healthy person's bloodstream in concentrations ranging from 4.5 to 5.0 m, while certain hormones may be present in nanograms per milliliter. Comprehending how this system works is essential to grasping life itself. Biomolecules are the essential components of all living organisms, and their quantities are essential for sustaining these systems. Biological systems must maintain a steady state, which is a non-equilibrium state, in order to function properly. As we know from physics, a system in equilibrium is unable to produce work. The continuous movement of biomolecules is a necessary condition for life, and an understanding of this concept helps explain the energy flux in biology. Biomolecules are essential for life to be maintained, and they play a pivotal role in metabolic pathways. These pathways enable organisms to utilize energy from nutrients and other sources, and they form the structural components of cells, transmit information and aid in the storage of energy. Without these components, organisms would not be able to maintain their living state. Metabolism is responsible for the continual input of energy that is necessary to maintain homeostasis in the non-equilibrium, steady state associated with the living state. Without an understanding of metabolic pathways and the biomolecules involved, it would be difficult to comprehend why the living state is a non-equilibrium, steady state. Biomolecules play an essential role in cellular metabolism, being the fundamental building blocks of life. Particularly relevant is ATP, an energy currency of the cell capable of being hydrolyzed to AMP, ADP and CTP, which supplies energy to metabolic processes. Glycolysis is a metabolic process that breaks down glucose to create energy. The process consists of 3 distinct phases, with the number of steps varying depending on the pathway utilized. Generally, 10 steps are recognized in the glycolysis process, each step resulting in the formation of a new compound, that collectively culminate in the formation of 2 molecules of pyruvate. The accepted values for glucose concentration of healthy humans are between 4.5 to 5.2 millimolar, with a range of 4.9 to 5.0 millimolar. This range is important to maintain for a healthy lifestyle and proper nutrition.